Hello students, welcome to PNYC. We are today going to study our chapter in Science 2, that is Introduction to Microbiology. So this happens to be the 7th chapter in your syllabi, and take it from me, it's going to be very interesting and it is slightly different from what all we have studied so far. So without wasting much time, let us begin with what all are we going to study in this chapter on your Introduction to Microbiology. So broadly, this chapter has been divided into two major parts, that is applied microbiology and the next is industrial microbiology. So a major chunk of it will be the industrial microbiology, which will be taking into consideration a lot of processes, production methods, which are utilized to make various products which are microbiological in origin, which most of us you might be knowing, and some of them it will be new enlightening for all of you. So without wasting much time, let us begin with it. So about industrial microbiology, it is divided into two types. So what we are going to study majorly is these two types. So the first is that production of various enzymes or proteinaceous substances from certain bacteria or fungi, that is through fermentation processes. This happens to be a first point. And the second most important point, which is actually found a lot of usage these days, is your waste management or garbage management. Basically, these are methods or methodologies to prevent your pollution. So, containment of pollution in any form, let it be air, water, sound, etc., through microbiological means, that is by utilizing fungi, bacteria, microbes, etc., is termed as microbial waste management, which happens to be in this broad category of industrial microbiology. So now in these two things we have to study a number of subtopics. So let us begin with them one by one. So let us begin with the applications to microbiology one by one. So here there are these 7 to 8 major categories that we can see, which we could term as applied microbiology or industrial. They are a mix of the two. Let us see one by one. So the first is your dairy production or dairy products. So what do you mean by dairy products? All that involves the use of milk, right? Starting from milk itself to its milk and applied products. Like you have got your curd, you have got yogurt, you have got cheese, so on and so forth. So basically, your dairy products, we'll be seeing them in detail for these three. So they are essentially influenced or obtained through the use of lactic acid bacteria, which we term as LABs. So this LAB that you see is a standard abbreviation for lactic acid bacteria. So when it comes to dairy products, the first these days in the market, which is a favorite boom in the industry, is your yogurt. All of us are aware of it. So basically, it is nothing but what we call in Marathi as dahi. So it is a different type of fermentation that is carried out, but essentially the mechanism remains the same. Now, depending on what strain of bacteria you use, you get those respective flavors. Now, apart from that, in the market these days, you get a lot of options to choose from. Blueberry yogurt, strawberry, banana, vanilla, so many flavors. Like you name them and there it is there. And of course, the companies, you people know better than us. So usually for yogurt or curdlings or fermentations, we use these two strains of bacteria. So this is Streptococcus thermophilus and this is Lactobacillus delbrueckii. So one is both of them are lactic acid bacteria, only the genus is different. So since I have written it in normal binomial nomenclature, I have underlined them. Otherwise, you can italicize but distinguish. So please have it a practice. It is a good practice if you learn. Of course, it depends on whether you take bio during your 11th and 12th, but nothing goes waste, right? So you can learn certain good habits from now itself. So it will be happy for you when you grow up in the next 2 years. So here there is a convention of underlining the genus and the species. So this S stands for the species, Streptococcus. This is your species. Similarly over here, this is your genus Lactobacilli or Lactobacillus and delbrueckii is your species. So you underline that. So as I told you, for curdling, that is yogurt, basically it is nothing but curd. We use these two strains. So if you see in our homes also, when your granny or your amma or your mother makes curd at night, she seeds the inoculum. So what is it? It is the pre-made dahi or the pre-made tak that she utilizes as a seed to the milk. So this is termed as inoculum in scientific terms. Inoculum is nothing but a suspension of bacteria and at times yeast. Mostly what we have is majority are bacteria, very few yeasts are there. Because yeast will give you a typical, pungent or, spoiled, stale odor. So the bacteria have to be there present in majority. So this inoculum is nothing but your seed which is going to curdle your milk overnight. What is curdling? It is nothing but aggregation or salting out or precipitating out of your proteins from it, which will give you the thick consistency, which is more pronounced in cheese. I'll let you know the details shortly. So after yogurt, the next most thing is utterly buttery delicious Amul. So we have our butter, which most of us like. So butter is of two types. It is the sweet cream and the cultured one. So cultured one is of course obtained through fermentation, wherein you separate out the fats. These days industrially, they simply defat the milk, and from fat it is processed industrially, and then you get your butter. But ideally, if you see in our homes, we'll first make the curd, and then from curd, slowly and steadily over days or a week of accumulation, we'll get butter. So that processes are getting extinct these days due to the modern lifestyle. But if some of you know, you are lucky enough. So this is about butter. Then coming to another very next popular product is cheese. These days we need cheese in everything, right? We need it in burgers, we need it in pizzas, we even need it in Maggi, and even our Indian dishes like dosas. So cheese production, initially when this was conceptualized, I mean cheese as a product. So cheese at that time was made using rennet or rennet. So what is this? This is a compound or an enzyme obtained from the mucosa of calves. Mucosa is your inner lining of the organs. Now, inner lining of which organs? It's of the stomach. So, cattle and calves are a part of the ruminant family. What are ruminants? Ruminants are animals who can regurgitate their food. So, they eat, they gulp down everything, and then they will pull it back, and then they chew, and you have typically seen cows graze, like all the grazers, in fact. Cows and buffaloes, you see, even if they are not feeding, if there is nothing in front, you still see that they are chewing something. So that is nothing but their regurgitated food. So these animals likewise are called as ruminants. Most of the cattle that we domesticate, including sheep, lamb, cattle, etc., are all ruminants. So these ruminants, their calves, their young ones, their offsprings, the fourth stomach, usually they have got four stomachs, I mean, not literally four stomachs, but they are like four compartments. So you can also call them as four stomachs. So from the fourth stomach, this compound or this enzyme is obtained, which is termed as renin or rennet. So in your textbooks, it's given as rennet. So please remember it likewise. So what happens is it leads to coagulation of proteins in your milk to form a hardened substance that is your cheese. So basically, it will add, those typical soury, buttery, and little bit of, bitter tasting qualities to your milk, to your coagulated proteins, which will give you cheese. So basically, in cheese, the process begins through fermentation itself. It is going to be fermentation of milk, your lactose to lactic acid, which will further aid the fermentation process. Apart from that, the proteins in milk, most predominant of which is your casein, or as we write the spelling over here. So this is your protein in milk that is present in major proportions. So casein hydrolyzate is again one product which is obtained from milk. So usually these whey powder, protein powders, if your gym grows, you must be knowing that they do contain casein hydrolyzate. So that is nothing but your readily absorbable protein which has been obtained from dairy source, that is milk. So during your cheese production, you need a seed. Seed means you need certain amount of bacteria which are built up as a consortium, which will carry out the fermentation process. And on certain occasions, there are also certain fungi that are used which add very typical flavors to it. Okay. So for now, what you can remember is a consortia consisting again of labs. Which labs? Such as these. So this is Lactobacillus cremoris, L. cremoris as I've written, L. lactis, that is Lactobacillus lactis, and your Streptococcus thermophilus, which we have even seen in yogurt production. So as I told you, the mechanism will be very similar to your yogurt formation, fermentation followed by curdling of milk, or precisely rather it's more simultaneous, and then what you get is your beautiful cheese. But cheese has to be aged a lot, right? So when you prepare yogurt or dahi, we also in Maharashtrian homes were aware of this product, which is favorite. You have it during New Year's typically, Amrakhand and Shrikhand. So what is that? That is basically even more hydrated dahi. So when you dehydrate dahi more, it gives you charka, as we call it in Marathi. So this is nothing but it's more of fat and less of water, which gives you this typical, smooth, smoothie-like and typical fat-rich consistency. So from which we make these two products, that is Shrikhanda and Amrakhanda. Now imagine if I further dehydrate them, what will I get? So when I do that, I go on removing the water molecules from it, right? So I'm dehydrating it. Now what will be left behind? It will be your proteins and fats. That is what precisely is somewhat similar to your cheese production. So in cheese, there is very, very little water. Now leave apart the exception of these cheese slices that you get in the market. Okay, they have a lot of water. You can actually fold them where they're very squishy and they even flip easily. They are like typical floppy. But classical cheese like cheddar cheese or the cheese bricks that you get, they are typically hard, right? So depending on how much aged it is, on what consortia it has been going through, the flavors and the price also of the cheese varies. So more the aged, more is the price. So is true for wines. Wines are also fermented. So more aged beverages are highly priced. The reason is that because it is believed that it is fat. Okay, it's not just a belief that storing them over periods, steeps in the finest of the flavors and gives you the typical aroma that one should expect through such beverages. Then after these dairy products, an allied dairy product, which is again very much into the market these days, and there is a lot of enlightenment about it, is your probiotics. Now, what are probiotics? Probiotics are nothing but basically your curd-like products. So, yogurt, probiotics are almost similar or one and the same. So, what do these probiotics do? It is a consortium of bacteria like lactic acid bacteria, LABs, that we saw. The most prominent one of them is this strain, that is Lactobacillus casei. If you see Yakult, okay, most of us are aware because that's a Japanese brand and there was a lot of marketing around a decade ago. And most of you at least once must have tasted Yakult. So what does Yakult contain? It contains this Lactobacillus casei shirota strain. So then after species, you've got some species var, etc., which is beyond your scope. Just remember it's Lactobacillus casei only. The strain was developed there, so it was named as shirota strain. So that is about Lactobacillus casei, again Lactobacillus acidophilus and bifidobacteria. So this is a different genus, but they also form a part of the probiotic consortia. Now here I have been using the term consortia. Then you would ask me, sir, what is consortia? So consortia is nothing but a group of organisms that are put together. Basically, it is a terminology utilized for our probiotics and prebiotics. So putting together certain yeasts, which are fungi, certain bacteria, and then making a consortium. Like if here you all were sitting, now you are giving me on YouTube. So if you are sitting in the class today right now in front of me, you would form a consortium, which is a group. So basically, these consortia, they are lab tested, they go through very stringent tests, and after a lot of effort, these probiotics come out. So probiotics are going to help in your gut health. They will help maintain the healthy bacteria population. The example given in your textbooks is of the Clostridium bacteria. So Clostridium bacteria again is a part of your normal flora. It is present in the lower end of the gut, and as a matter of fact, after you die, okay, after you're physiologically dead, it is the first organism to invade your body. It means it will, spread into your tissues. Basically, until you're alive, see, we all have got bacteria on our skin, on our guts, on our bellies. You know that half of our poop is bacteria, that's a fact. But they don't invade us, right? They don't eat us. They eat what we feed them or other food we eat. So when you die, they are opportunistic, most of them. Some of them will die, some of them will survive by penetrating you. So Clostridium is one such species, hence it's also known as an opportunistic pathogen. So these consortia of probiotic bacteria help to keep such opportunistic pathogens in check, or rather by preventing their growth, in some way inhibiting their growth, or by killing them. So a lot of lactic acid bacteria have got 2-3 things which help to keep their population in check. So first is their self-population. If you are too much in number, if this classroom was occupied, and there was no place for you to come, would you be able to come? No. But then of course you have this option of PNYC YouTube biology videos. So then that is one strategy. You would not be able to grow because you already have a lot of population, as we studied it in our first chapter, interspecific competition. That is one part. Second thing is, the name itself justifies. They are lactic acid bacteria. So they are going to produce a lot of lactic acid, which is going to reduce your pH. So when the conditions are acidic, we know many of the organisms cannot survive. So the pH, it is a physical property. So apart from chemical properties, they'll be having this physical property, because of which the population of Clostridia will be kept in check. The third important thing is, some of them do produce certain antibiotics or bacteriocides. So what are these compounds? This also most of you must be aware of. So antibiotics are compounds which are produced by certain group of bacteria itself to kill the other bacteria. So the bacteria which produce such antibiotics are usually predatory bacteria like actinomycetes. A large amount of antibiotics that we use today have been obtained from actinomycete species. And then of course the most famous penicillin during the Second World War. It was from Penicillium notatum. And there is one more species. So Penicillium is a fungi, of course, but your lactobacilli are bacteria, of course. So due to a number of physical and chemical factors, they will be keeping the pathogens at bay and check, which terms them as your probiotics. So they are important. So even when I'll give you one more example. When you have a stomach upset, what do? Of course, you stop motions by taking a lot of medication, allopathy, Ayurveda, or home remedies. And then what are you advised? Think. You are advised to eat curd, right? Or fermented products. What is the reason? This is the reason. Because they contain this consortium, which is going to help repopulate your gut with these healthy bacteria, right? Like Lactobacillus casei, Acidophilus, then you have your lactis, so on and so forth. A lot of species are known which act as probiotics. After that, the most famous confectionery products, which all of us, are relishing on almost every day, is your breads, croissants, pastries, khalis, whatnot. So what are all of these? They are all maida products, refined flour of wheat. So this also is kneaded into a dough and fermented. So they don't immediately roll out breads, right? That we do with our chapati bhakri. We just knead the dough and then we roll them out. And then we bake them, heat them, and they're ready. But with breads, they are left aside for fermentation. Now what ferments them? We have to give the seed. Now in this case, the seed will be your yeast or your fungi. Now which yeast will you give? It will be your Saccharomyces cerevisiae, or as I have abbreviated it here as S. cerevisiae. Saccharomyces species is a genus of yeast which has been for centuries now used in fermentation. So for breads, typically you use this organism, and the mechanism over here is similar. When you add yeast to the dough, the yeast is going to utilize the sugars, the proteins, and the other essential minerals and fine chemicals present in your source, that is your refined flour or maida as we call it, and cause the production of two major products. What are these? It will be your CO2, that is gas, which will escape, and the second is ethanol to a certain extent, and also acid if depending on what species it is. So mostly yeast are known to produce alcohol and carbon dioxide, but to a certain extent they may also produce acid depending on what conditions they are subject to and what species or what strain of yeast are using. So then a little bit of extra knowledge for you people if you're interested. So depending on what yeast it is being used in the fermentation, you have got top yeast and bottom yeast. So basically this is a concept for wine production, okay, not for bakery. So depending on whether the yeast grows on the top or it grows at the bottom in the submerged condition, you get different products, different flavors of wine, different flavors of vodka, etc. So that is about your use of yeast or fungi for fermentation processes. Coming to next, it is vinegar. So vinegar also is a very valuable product that has been found to be utilized in a number of food products for preservation. So what is vinegar? Vinegar is nothing but your 4% solution of acetic acid. We know all what is acetic acid? CH3COOH. In your chemistry, you must have studied. This is acetic acid. So a 4% solution of acetic acid in water gives you vinegar. So vinegar is used as a preservative in your pickles, jams, jellies, all the industrial pickles that you get, okay, these days, it will contain vinegar. If you read the labels, you will be aware. There is sodium benzoate and the second product is vinegar. Even if they don't clearly write it's vinegar, you see there are certain annotations given, ENS2A, ENS474. So all of these are compounds, but they have got their industrial nomenclature. So they are named that way, but vinegar is a must component when it comes to preservation. Then, apart from this, they are also used to cure certain ecological aspects or help in farming, help in maintaining the homeostasis in our ecology. How? That is by use of biopesticides and for nitrogen fixation. Now, you would ask me, why do we need nitrogen fixation? We already have 70% around in the air. No dear, but that is your atmospheric nitrogen. It is biologically unavailable. Now, if I tell that your DNA contains nitrogen, now can I just swallow air and that nitrogen be absorbed into my synthesis pathway? That isn't simply possible, right? So, when we talk about nitrogen fixation, it is with reference to auxotrophs. Auxotrophs as in plants are primary producers. So, some of them have got symbiotic bacteria residing in their root nodules or rhizomes. Hence, they are termed as rhizobacteria. Now, the free living ones are the Azotobacter species, as I have rightly written in the parenthesis over there. So these bacteria have the capability, Azotobacter and certain blue-green algae as well, that is termed as PGA. They have got the ability to fix the atmospheric nitrogen into biologically available nitrogen as nitrates or nitrites. So there is a typical very long pathway which we study in biology, but not for your standard, in which you understand that how atmospheric nitrogen, which was gaseous in nature, has been fixed into the form of nitrates or nitrates, which are conjugated to certain metals. So they form minerals later on, like sodium nitrate or sodium nitrate, so on and so forth. And that is how this nitrate makes the nitrogen biologically available or bioavailable to the plant. Then coming to biopesticides, an interesting aspect, a lot of research is being done on it, and there are like so many research papers also. The government is also funding for this because you see these days the chemical pesticides or insecticides that we are using, there is a lot of resistance acquired to them. So we are now turning to Mother Nature to see how this interspecific competition has actually helped to keep the predator-prey relationship, in homeostasis. Now this can be understood with the example of probiotics. What did I tell you? This consortia helps to keep in check what? Your pathogen. Now the pathogen happens to be Clostridium. So here also, what I mean by pests is your pests on the crop, which could be your caterpillars, your wood borers, your stem borers. So naturally they have got certain predators, predatory organisms, which could be insects, worms, birds, they could be anything. But now, since we are studying microbiology, we typically understand which microbes are being utilized. So a number of microbes are known to act as biopesticides or biocontrol agents. The recent study, a year ago, if I'm not wrong, in which they had introduced male mosquitoes which had Wolbachia in their guts or in their body. So Wolbachia is a species of bacteria. Okay. So they were infected in the labs. These mosquitoes were bred and they were infected with Wolbachia bacteria. So what happens is when Wolbachia is infected to male mosquitoes, they are unable to mate, and that leads to a control of the population. So a lab study was done on a pilot scale, and of course after certain refinements they were let out. I forgot the region, but somewhere in Southeast Asia wherein there's a huge nuisance of mosquitoes. We have them all over, but there it is more pronounced. So these Wolbachia infected mosquitoes were intentionally released into the environment. Now why? Because it was presumed that they will try and find mates, and then it will lead to ceasing of their species. Like it will lead to a death, eventual. Now, extinction is a big term to use, but for now, I would say like birth control for mosquitoes. So even if they breed, they will be not bearing offsprings like eggs. They will not be able to lay, or most probably they will be dying. And then the other advantage of having Wolbachia is it has got certain roles to play in vector-borne infections by preventing them. What do I mean by vector-borne infections? You understand malaria? Everybody is aware, especially our parental generation. They have had a lot of, even today if you tell them they, get chills or probably nightmares because they have seen people pass out with malaria. Now, these days due to our living conditions, improved, conditions of ventilation, we have got these nets around which you put on windows. We have got all out and whatnot. All these anti-mosquito coils that you get. So in olden times, this was not much pronounced. It was not that much available, which led to a lot of mosquito bites. Now, if your mosquito harbors the parasite, then it leads to malaria, right? So these Wolbachia infected mosquitoes are not only compromised on their breeding potential, but they also help to kill the parasite of malaria that is present in the organs at certain stage of their development. You know that malaria parasite has got two hosts. It has got humans and it has got the mosquito, the female mosquito. So when the female Anopheles mosquito will bite you, it will let in the chytron, the pirozoites into your bloodstream. They will develop and they will be passed on to the mosquito, and then they will be passed into the water, and that is how they will be completing their life cycle. This is extra, you need not remember. I'm just telling it out of interest that how ultimately microbes can be used as biopesticides. Then when it comes to crops, certain enzymes from bacteria, especially if you know about BT brinjal or BT cotton. So there is a bacterium which is known as Bacillus thuringiensis. I write it over here. I hope you can see it. So it is Bacillus. So this bacteria releases a toxin, okay, which is known as BT toxin. Now this BT toxin actually gets activated in the alkaline guts of the caterpillars that feed on your crop. So their gut is alkaline. Alkaline as in pH above 7. So when the bacteria feeds on this crop, this toxin gets activated into their intestines or in their gut. Now when this happens, it leads to perforation of their intestine, which means it will drill holes, it will make injuries. So due to the leakage or a leaky gut, the caterpillar dies. So it dies a very painless death without understanding. So that is about BT cotton, BT brinjal. So this is also a wonderful example and a successful one of your biopesticides. Now, apart from this, they are used in the second major aspect, that is for waste management. So let us see that. Just give me some time to prepare for it. So coming back to our topic on waste management or garbage management, there are these four major types which I have divided them into. And apart from that, there is one small aspect of your introduction to microbiology in which these two are also major uses of microbes, but they are nonetheless, apart from the second one, that is use of microbial enzymes. Microbes for fuel is not much pronounced yet. I mean, in degrees or magnitude in which it should have been. Now, of course, there are a lot of limitations to it. The one and the most common is your methane production, and your second is alcohol or ethanol production. So microbes are used as fuels. Now you would ask me, sir, why are you terming ethanol and methane and hydrogen gases as fuels? So hydrogen fuels, these are the next generation fuels that we're talking about. So now we are talking about hydrogen fuel cars. Hydrogen is such an abundant gas on the face of Earth, but nonetheless, it's extremely flammable and moreover explosive. Like it can burn, explode. It can burn exploding. So it's very difficult to compress hydrogen and keep it going to the safety issues, so on and so forth. Like even for LPG and CNG cars to come into being, that there has been a lot of research. So hydrogen is even more pressurized than CNG and even more deadly or more dangerous to handle. That is why in certain parts of the US, these big companies, they are having trials on these hydrogen fuel cars. But nonetheless, for them to hit the roads, it will take still a few decades, I believe. So hydrogen as a fuel is used by, production of hydrogen as a fuel. For this, we utilize your anaerobic bacteria. What do I mean by anaerobic bacteria? So basically, they are these bacteria which grow in anoxic conditions. So you see, all of us are breathing right now. I am breathing, you are breathing. So this is due to what? This is due to the presence of oxygen in the air. Now you have certain bacteria or certain microbes. When I say microbes, I also include fungi to the list. So fungi, then bacteria, and certain other unicellular prokaryotic or eukaryotic organisms. They are able to sustain zero oxygen conditions. Rather, they are obligately zero oxygen requiring, which means you give them oxygen, they will die. It is toxic to them. So the best example, you remember I told you about Clostridium. So Clostridium is an obligate anaerobe. Obligate means compulsory or strict, mandatory. And anaerobe means any organism that grows under anaerobic conditions. Aerobic meaning oxygen and anaerobic no oxygen. Remember it this way. So anaerobic means there is no oxygen. They don't require oxygen. Now this is also due to a presence of the respiratory pathways. They lack the enzymes which can utilize oxygen, which makes it toxic. So what do? We utilize this for treatment. So whenever a person is being suspected to be affected with tetanus or tetanus as we call it. Now what is tetanus? It is Clostridium tetani. So it is again a Clostridium species. So you know, if you have injuries on the leg or the hand, what is done is your hand or legs are enclosed in a chamber and oxygen is pumped. So oxygen is fueled through a pipe and it is maintained under high pressure. So hyperbaric oxygen theory, that is a treatment modality to treat tetanus or Clostridium or sepsis affected patients. Not deviating much. So microbes as fuels, you can utilize such organisms that grow under anaerobic conditions and give you hydrogen and methane as your byproducts. So they are going to grow and utilize all the food that you give to them, your molasses, your farm waste, your saw, hay. So these are all what? Your farm products. Best example to isolate and obtain methane producing bacteria is your dung. Cow dung, cattle dung, horse, camel, etc. Predominantly it's cow because that's the most farmed cattle world over. So basically you see biogas fermentation plants, what do? They use cow dung as the seed. Now why is that as a seed? Because it contains a lot of consortia or methanogenic bacteria or methane producing bacteria. So they are going to act as a seed, and as I said, when it is produced, it is kept covered as a dome. There is a nice picture illustration in your textbooks. Please go through it. So that maintains an anoxic, not like 100%, but more near to 100%, I would say. So now what happens is your methane production starts because bacteria at the bottom. Like imagine this room, the height of this room is around 10 to 12 feet. So in the top 1 to 1.5 feet layer, if there is oxygen and it is completely filled with sludge and mud, what would happen? The layer, like where my feet are or where my hands are right now, it would be still anoxic because oxygen is not going to diffuse or penetrate down there. That is what happens during anaerobic fermentations. So through anaerobic fermentations of certain organisms, and the examples are Methanosarcina, Methanobacter. So these bacteria are going to give you your methane and hydrogen as fuels. Okay. Then comes the third one, that is a liquid state fuel, which is your ethanol. Now ethanol in large amounts is obtained through yeast and fungi through fermentation of molasses. Molasses are, as we know, are a waste product of your sugar industry. So whenever you have cane sugar and jaggery that is produced, the liquid slurry that falls out after the process is known as your molasses. So depending on what waste product you use, what strain you're using, you can define what type of product you would expect. Will it be a mixed acid fermentation? So along with ethanol, you can also get acetic acid. Or it will be a sole ethanol fermentation. It means you will exclusively get only ethanol or ethyl alcohol. There are few tables given in your textbook. Please don't overlook them. They are a beautiful consolidation of the organisms, their use, and what do you get from them, like in terms of flavors and all. So there are 2-3 tables in the textbooks. I hope you must have seen them. If not, please go through it. So that is about your uses of microbes for fuel and other flavoring substances, this and that. Apart from that, a major portion of microbial products that we use in today's world or today's date is our microbial enzymes. So microbial enzymes, what are they? There are a number of microbial enzymes that we use. We use them in the industry, we use them for medicine. So what are they? They are your lipases. What are lipases? Lipases comes from the word lipids. So anything that digests your lipids or fatty acids are termed as lipases. Then you have got your proteases. So proteases are what? Coming from the word protein, derived from the word protein. Anything that degrades your proteins into what? Peptides or amino acids are termed as your proteases. Then you have got lyases, hydrolysases, hydroxylases, so on and so forth. So many of them, like I could name, and it would be tomorrow's morning or an entire day. So these are your industrially and clinically important enzymes that you use. So whenever you have had a lot of food, let's say you go out with friends on a party or you go to a restaurant. So when you eat more food, you need something to digest, right? So the typical advertisements are of Eno and all, right? Eno, Jaljeera, whatnot, whatever are used as appetizers. But apart from that, external enzymatic aids are also used. Now, what do I mean by aids? These are these enzyme tablets that you get in the market. So they contain a enzyme called as fungal diastases. I'll write it here. So fungal diastases are derived from Aspergillus species, if I'm not wrong, or certain other fungi species which give you this enzyme. Now this enzyme is going to help you in your digestion of complex matters. We eat complex fats, right? Sorry, complex foods. We eat carbohydrates. Whenever you go to, you just eat sugar, table sugar or cane sugar? No, right? You'll have chapatis, you'll have parathas, you'll have different rice, salads, samosas, whatnot. So all of them are essentially coming from your complex group of carbohydrates, right? Simple carbohydrates are sugars. So you need fungal diastases, pepsins, etc. to digest the food. Amylase, a very good and excellent example. It is going to digest your starch that is present even in your saliva. There is a small test that you can do. You can chew the bread or chapati for long in your mouth, like 30 to 40 times, whatever. After a while, you will start feeling it sweet. Why is it sweet? Because the enzyme amylase is present in your saliva as well. So when this is secreted, it is going to break down the starch present in your roti, chapati, etc. because they are made up of wheat or jowar or any other thing that you might be using. And it is going to break it down into simple carbohydrates, that is sugars. So your amylose or amylopectin that was there, not amylopectin that's there in mammals. So amylase basically, since we are talking about plant originated foods, it is going to be broken down into your sugars. So they are amylases. So on and so forth are the uses of microbial enzymes for day-to-day use in the industry as well as the clinical aspect of it. In industry, a lot of proteases are used, lipases are used, even in the tanning industry, the leather industry, to remove certain, when you have this skin dehydrating process, you just can't butcher an animal and get leather out of it and just skin it and you have the product. No, that's not possible. So a lot of treatment it goes through. So certain sophisticated ones do utilize these proteases. Even for preparation of these probiotics or prebiotics, and of course the other biological really relevant products, you utilize microbial enzymes on a large scale. Then coming to the interesting point of waste management in your chapter, we have these 4 major points. What are they? First is clearing of the oil spills. Basically, this happens in your seas, open seas, where oil is transported. If you don't have a pipeline, oil is going to be, extracted from the ocean floor. You will have ocean taps as in not literal taps. They're like huge pipes which pump out the oil from the ocean floor wherever it is found, and then that is transported through cargoes or vessels to the respective nations. So when there are accidents, there are tornadoes, there are hurricanes, whatnot. The sea is very rough out there. So you have these vessels which turn upside down, they flip. There are accidents, and then what happens is this extracted oil spills. When this spills over the ocean, we know that the density of oil is less than water, so it is going to float, and that is a huge mess. And huge as in how much it can spread to hundreds of kilometers in the ocean. And mind me, students, it creates a havoc on the ocean life. The birds die because their feathers are oil wet. They're oil sunken. They can't fly. Their feathers don't dry out because now they are wet and they have an oil level. So fish cannot breathe. Oxygen does not penetrate the water. So much. Like you could Google out or see a few YouTube videos upon oil spills to see how serious it is. So for clearing out such oil spills, what do you do? Clearing out as in, I want to fill up the oil? That's practically impossible. Nobody does that. So what you do is you put this powder or you spray this consortium of bacteria, that is this Nocardia species and Alcambora. So these organisms are known to degrade oil. Now what is this? I gave you a clue over there. Lipases. So these are enzymes that they will produce in pronounced amounts or in high amounts, which will help to digest or clear the oil. Basically by utilizing them. So they will be utilizing these hydrocarbons which will degrade the oil. And one more point, they will not be secreting lipase. Lipase is basically for lipids which are of biological origin. This is mineral oil. So they will have another mechanism. Of course, they may secrete lipases as well. But the mechanism and the pathways or enzymes are still distinct for the bacteria that we use for clearing oil spills. Then our second point is heavy metal removal. A lot of bacteria have the ability to quench or to chelate certain metal ions. What do I mean by these two terms? It is you sequester them. So when you have got heavy metals, tell me for example of heavy metals if you can. The best is that is present in the thermometer, hydrargon or mercury, Hg. Then you have cadmium, you have lead, okay, so on and so forth. You can add on to the list. So when microbes are utilized to quench or chelate these heavy metals from the environment, they are going to be termed as waste managers or organisms that are used to clear out heavy metals. So basically, heavy metal pollution is a serious issue in certain rivers where industrial effluents are just left untreated into the rivers. So that is due to a low quality of stringency that the company follows, or it is carelessness. Careless attitude on their part, wherein they say that we care no less and they just let out the pollutants in the open water bodies. So microbes can be utilized for this. Apart from that, a serious concern is the sulfur removal. So there are certain chemoautotrophs, bacteria that utilize chemicals instead of carbon for their survival. Now these are termed as PSBs or purple sulfur bacteria or green sulfur bacteria. Typically, they are abbreviated as, just as we had lactic acid bacteria, similarly you have got PSBs over here and GSBs. Purple sulfur bacteria and green sulfur bacteria. So they are going to utilize sulfur for their growth. Now you have something which is to be removed. You want to get rid of it. So what do? You get somebody who utilizes it. So that is what exactly they are doing out here. You use PSBs and GSBs in certain occasions to remove sulfur from the environment or from the polluted water body. Then comes the most important point under our topic, that is plastic degradation. I need not reiterate. Given a choice, I would skip this. Everybody, literate, illiterate, every one of us know how plastics have gone far and wide into our environments, into our ecosystem, right up till our blood. I challenge you, if you go to the clinic and take a blood sample of yours and do a microplastic test, it will be positive. All of us by now have got microplastics in our body. So microplastics are again that you can't see. You will not see your mobile or pen covers and pen cases in your bloodstream. That is impossible. But then microplastics are your filaments, fibers of plastic, which you might have ingested accidentally or involuntarily while breathing or eating something, which are there in your body permanently residing for now. So they are your microplastics. A serious concern and issue. So what do? We have to clear them as well. So here again your friendly bacteria come to your disposal. They are your Nocardia species and your actinomycin species. So both of these are a part of this HCB consortia. HCB is your hydrocarbon clastic bacteria. Hydrocarbons, plastics are hydrocarbons, right? Basically alkenes. You have studied your chemistry well, I presume. And they are going to, clastic means anything that lyses or breaks the thing. So your hydrocarbon clastic bacteria are going to degrade your plastics. Of course, they have their own mechanisms which you need not study. You can just remember the examples from now. For now, that is Nocardia and actinomycins. So, classical example of plastic is your PET bottles, your PET bags, polys, whatnot. So, what is PET? PET is nothing but polyethylene terphthalate polyester. So, it is an ester of an alkene, hence we term it as PET. That is nothing but polyethylene terphthalate polyester. So, certain bacteria have now been known to lyse and degrade plastic, but that is at a much slower rate. And let me tell you something which is very personal. Every microbiologist thinks that anything present on this phase of earth will be degraded by bacteria one day. Okay, but that is, I won't say that is impossible. There is always room for hope and hope. But you should not be overtly optimistic about certain things. It doesn't mean that Mother Nature will keep clearing your mess and you just keep on producing and polluting her. That is not the case. So there are certain plastics these days that you get. Typically the black bags that you put into your waste bins at your home. Have you seen them? I hope you have. Then if you have been at a nursery, the saplings that you get, they are in those typical black bags, right? So the cylindrical versions of them. Ever wondered why are they black and why are they plastic and what plastic is that? So that is nothing but your PLA. PLA is your polylactic acid. So it is a polymer of lactic acid which is a plastic and it is biodegradable. So that is why you use PLA bags for this purpose. So with this we come to an end of your chapter on Introduction to Microbiology. Hope you have enjoyed this and see you soon with the next chapters in your syllabus. Till then, goodbye.